Hello and welcome to our new podcast episode of Relukata. I'm your host Vakas. Hope you are all well and keeping well. Keep practicing the guidelines of social distancing and keep washing your hands as much as possible for you. I will start by telling you that Relukata is a cricket podcast. However, as you know, there is not much cricket going on. Uh, and it is an extremely extraordinary situation with the virus spreading at a very fast rate in most parts of the world. So our thoughts go out to the people who have lost their loved ones and people who are suffering from this disease. So in this difficult time, we have arranged this episode to talk about something that is of great value to all of us, that is related to coronavirus testing. So one of our Relukata guys, Shulolit Banerjee, he has been part of a team that has been working on the development of a new coronavirus test. And this, with the development of this test and some other test methods, they can give us control over the spread of the virus. So the scientists and the experts all over the world, they have been stressing about the importance of the tests to flatten the curve, so to say, before a vaccine becomes available. However, for the traditional PCRRT tests that have been used worldwide right now, the reagents or the chemicals that are used for these tests, they are in short supply. So there is a need for newer test methods to be developed. And so such a test, it seems that it has been made possible by the efforts from Shulolit and his colleagues at Norway's Teknisk Naturvetenskapelige Universitet, or simply called NTNU at Trondheim. So therefore, I will introduce Shulolit here. So Shulolit is basically from Calcutta, Bengal in India. And he has done his undergraduate studies from there, from Bengal. And since then, he has moved to Norway and started his master's studies first there, if I am right. And then he has gone on to proceed with his PhD studies in chemical engineering at the same university. And now he's a postdoctoral researcher at the same university. So I will ask Shulolit to introduce himself a little bit. Shulolit.
Yeah, thank you, Akash. I think you have already told everything that needs to be introduced. But to add to that, what I would like to stress is that I am also working as a researcher at TU Delft in the Netherlands. And actually, what we have developed in this test kit is a part of the work or mostly connected to the project of TU Delft, although I'm working on it in Norway. So that is the only additional thing that I would like to mention about myself and the work.
Okay, right. I will take this opportunity to introduce the other participants. We have Asif Javed Dillon, who is also a postdoctoral researcher in chemical engineering in Sweden. Say hello, Asif.
Hi, this is Asif.
And then we have Atif Irshad, who is speaking to us from Lahore. He's also a mechanical engineer. Say hello, Atif.
Hello everyone. Hope everyone is fine.
Yeah. So, so now we will start with the serious discussion about the test that Sulolit and his team has developed. So, Sulolit, tell us about this test. How does it work?
Yes. So before we start discussing more about the test, I would give a bit of a preview of how we got involved into this work. So it was a request from a department of circulation and medical imaging at NTNU, which is located in the same campus as that of the university hospital, who wanted to check if there was anybody at NTNU who is capable of synthesizing some magnetic beads. That was all the request we got. And what we were asked from up above, basically the heads of the groups, is that the request was forwarded to me because they knew that we are working with such magnetic particles for the Delft project. And they told us that if this is possible, then please start the work. That was all. And then it was 3-4 days of hard work from me and a PhD student working with me, Anuvansh. And then came in a couple of other colleagues from our department. And then we formed a team of 3 mostly, who kind of led this process at our side. And what happened in the time being is that there were 4 other people involved from the Department of Medical and Circular, or from the Medical Imaging Department at NTNU. And there was one more person involved from Sentulav's hospital, which is the requesting authority for this particular test. And in the first 3 days, we started testing. We started producing in small amounts. We started testing those samples with the standard tests at the hospital and the Department of Medical Imaging. And then we started getting some promising results. And actually, our beads started to perform better than the commercial ones, which were actually in short supply. So just to add to what Wakas was saying is that this is not a different kind of detection method. It is still a PCR-based detection, but the chemicals are kind of the reagents that we use are homemade. So it is something that can be easily made without being dependent on some of the scarce chemicals in this crisis time. And what is also important is that, as I will mention later on when we come more into the process, how it works, is that it doesn't require any fancy equipments on the production side. And then it's a standard, the test can be performed in standard qPCR setups. So that's kind of the preview of how we got involved into this. And then I would tell you a bit more about the test, maybe. So the way the test works is that we have these magnetic beads, which are mixed with a buffer, a lysis buffer, that opens up the virus. And once the virus is opened, since this is an RNA virus, so that RNA is extracted or isolated from the virus, and this binds to our beads in certain conditions. And then we, by tweaking some existing protocols at the hospital, we release this RNA in water, which is then detected using the qPCR to find if the patient should be tested positive or negative. So that's kind of in a nutshell how this test works. And what our part in this whole project is then to provide them with these beads.
Mm-hmm. Okay. So that's quite interesting. So how do you think this test can benefit in the current situation if there are already tests that work? So why do we need these tests specifically?
Yeah. So I could first start from a very local perspective, then go to a more Norwegian perspective, and then finally to an international perspective. So locally, St. Olav's Hospital couldn't get kits from commercial suppliers because of the fact that there is a scarcity, there is a long waiting time, and also since there are so many other countries with a much affected population, some of the kits are strategically directed to those countries. So there was a shortage, acute shortage of kits as we talk 3 weeks back from now. And then with the validation of this method, so we first validated it in the labs and then validated with actual patient samples and found out that these methods, our method is more sensitive than the methods commercially available. Because we home grow our magnetic beads and also tweak our buffers to make them more specific to this COVID virus. So what then happened quickly is that St. Olav's Hospital took this process into their regular process and ran it side by side with their commercial process because it requires some validation from the health agency to be applied in hospitals. And that's what we are now in the process of getting clearance from the Norwegian health agency to allow us to be used not only in St. Olav's, but also in other hospitals in Norway. And this is what we are now working on, is to then produce beads for tests that could range between 300,000 to 500,000. So as I mentioned that when we started using or producing small samples, which could do 1,000 tests. From there, in a matter of 1 week, we have gone down to a production capacity, which is only, as I see it, possible with a chemical engineering background to produce close to 60,000 tests a day. So that's what we are able to deliver now, but this is much higher than what is required right now locally and maybe also within Norway when we get the numbers. And that opens up for international distribution of these tests or involvement or collaboration. We are ready to kind of help any country that requires assistance, but it has to be kind of a bit regulated since some part of the protocol is not to be disclosed to besides the ones who would like to use it. So yeah, there is a little bit of, I won't say there is no problem in this method being used for the detection of COVID all around the world, and we are in a situation where we can provide that under all costs as a service to other countries, but with some legal, I think it's called licensing or something of that sort, for use for non-commercial purposes. That's what we are able to provide right now.
Okay.
Okay, I've got a question. Yeah, I've got a question, Mr. Banerjee.
Yes.
So you're saying that with this scheme of testing, you will be able to ramp up that, which is what most countries want. But you are also saying that it's not possible. There are some legal implications. So is it possible that you might in future collaborate with WHO?
Yeah, coming to the legal side is, the legal side is only that different health authorities have different approval methods in different countries. So that's not standardized internationally. So it could be a very, the way it has now been approved, once it is approved by the Norwegian Health Agency, and once it starts to be used on a larger number of patient cases, it also helps us to use this to leverage other international organizations, including WHO, to adapt this method in whichever form it's most applicable to a country. So yes, I mean, I am very, I mean, we as a team, me and uh, uh, Professor Magner Puris from the Department of Medical Imaging, who are leading this team, we are very much interested in getting involved with countries that require this to do mass scale testing. Uh, and what I didn't mention is that our method, since it's more sensitive, it can detect low viral loads. So it can do an early screening if that makes sense for several countries to kind of screen a certain section of the population, find out that they are not COVID positive, and then allow them to go back to work slowly. So
Okay.
yeah, that is kind of what I see currently for these protocols.
Yeah. I have a question like when you scale it up, so what about the reagents for this test? Like how large quantity is available that they will not got short at like for the other test?
Yeah, very good question. I mean, so the process consists of two parts. One part is the bead production, whose chemicals are very easily available. I mean, it is not a problem, common chemicals. On the other hand, the buffer, which the Department of Medical Imaging is producing, that consists of some chemicals which are required, which could be the limiting case for these methods. From Norwegian perspective, we have got enough supplies to run for several thousands of tests, and there is more in the production. I mean, in the purchase. So that chemical is not a problem. It's only that that chemical is not being supplied by the major commercial suppliers right now, and most of it is coming from China. So besides that, I think the reagents are not a problem. What could be a problem in mass scale testing is plastic. The swabs which we are using to take the first sample from the patient, this could be a shortage, and things like that. So the reagents at this point, they are let's say yellow on a risk assessment, whereas the other plastic materials could slowly go into red if they are not supplied worldwide. So from a Norwegian scenario, I think there is this committee which is working on it, an expert committee, trying to risk assess what is the backlog. I mean, what are the critical weakness points in this whole development protocol, or getting it from the labs to actuality. And what we find now is that our contribution is actually between yellow and green. So the reagents is not a scarcity at this point.
Yeah, but sorry, but this you as you mentioned this plastic. So is this specific for this test, or it's the same for the other test as well?
So in a general qPCR, yeah, it's the same for other kinds of tests as well, other PCR based tests. But whether a part of it could be done manually. But for example, in most of the countries, they have automated this process, which requires special kind of plastic. But we have made the protocol available both to be used manually and using automation. So in order keeping this in mind, that plastic required for specific instruments that run this test could be a challenge in the near future.
Yeah, I have one more question. Can you elaborate more about these beads, like how they are made or what it is actually?
I could only say that these are magnetic, and they are, those are the magnetic beads.
Nanoparticles, maybe you can.
They are composed of nanoparticles. Nanoparticles, yes.
Okay.
Yeah, that's all I can say for the time being. Yes.
Yeah. Okay. Yeah. Thank you.
Okay, so Lalit, we know that in order for the test to be approved to be used among humans, it has to require a lot of testing. So how much patient sample testing have you done?
Yeah. So in Trondheim, there have been 100 patient samples. Some of the samples were difficult to also determine using the existing uh, routine practices in the hospital, but our tests were able to give very good results for those. We have done 100 so far at the hospital. There are several which are being tested, so we will get the data in as the time progresses. But they're being tested in other hospitals in Norway. It is, I mean, in extraordinary times like this, since this is more of a, it is not injected into the body of the patient, so it's a clinical test. So in such situations, I think the health agency is setting up a bit of a different approval policies within small scale production. So I'm not very aware of how that works, but I am only informed that this would be not it wouldn't take so much time that people are starting to not getting tested because the health agency cannot give an approval. So there should be enough number of tests done, as you correctly mentioned, Wakas, but not as rigid as some of the other tests which are commercial, because this is completely done so far for a non-commercial purpose to help screen out more people and meet the challenges currently.
One thing that I have gathered from what you have been describing is that the amount of time that it takes for the test results to arrive. How much time does it generally take for the results to arrive, and can this be a problem going forward going forward? Because if the tests start jumbling up and people have to, get the results, and maybe this all this analysis, it becomes too much.
Yeah, I guess at this point, the time that you take a sample from a patient and then you get the results in the hospital with this protocol is between 2 to 3 hours. So it's not a long waiting time. But then, as you correctly mentioned, that if there are like 100,000 patients being tested a week, then it's also a lot of data. And I'm aware that some let's say people who are more efficient in IT and computer science are working to make this possible. So there is some work going on in that regard to kind of register the samples, get those quickly to the labs, and get the data. So the test itself doesn't take much time, to be honest. But the challenge is, as you know, with all these, getting the sample, properly storing them, bringing to the lab, running. So at now, the hospital in Trondheim has a capacity of running 5,000 tests a day. And I think they're running very little tests right now because we are all waiting for kind of these approval coming through so that it can be used on a large scale. So the time to test is quite low, but data, that would be would be a challenge at some point when there are 100,000 people getting tested.
Yeah, I can imagine that. Okay, I have one more question about the patenting and licensing procedure for this going forward, since as you said yourself that probably your lab might not be able to meet up with all the workload. So maybe it has to be you have to license it to some other lab. So have you guys been thinking about that?
Yes, I mean this once it came out in the Norwegian media, we have been contacted by people from, to be honest, all over the world, from Ireland to Uganda to several other countries who are India, Qatar, trying to see if this product can be used and if they would like to market it. So the day the news was published, the next day we were already in meeting with the technology transfer office at NTNU to understand how should we answer questions and how should we go about. So we haven't come to a it's not an easy decision to make because there are ethical issues around it, there are commercial issues around it, and there are, of course, the most important thing is the current crisis situation. So having taken all into account, what we have done so far is we have filed a patent for the process, but we are thinking of developing licenses to people or to organizations who can run this. The only challenge that we have so far figured out is the transfer of technical competence required in some parts of the process. So that is something we have been discussing back and forth to see how that might be possible to be used in other countries. And we are not yet sure of what the strategy should be. We are still discussing to find out the best strategy. And again, to make it clear, we are not in a, or we are not inclined to make a commercial benefit out of the situation, but we want at the same time to protect the misuse of this discovery by other organizations or individuals, if that can be the case. So that's the only drawback, let's say. But we are discussing about licensing, and we think that different kind of licenses for different kinds of organizations might solve and might help humanity in this situation. So it's on the agenda, but we still don't have a very clear strategy, and we are still discussing with TTO and other involved people to understand what's the best option. And we hope to arrive at a decision once by the middle of this week, actually.
Oh, that's nice.
Yeah.
So, yes, Asif, if you have any question.
No. Carry on.
Yeah. So it might be that you and your research group have to train some other people to set up some new labs in some other countries, which frankly most of the countries are in desperate need at the moment to have more capacity to do as many tests as possible. So I will take this discussion to our own countries where we originally come from, Pakistan and India. Do you think it might be possible for people of our country to benefit from this? For instance, if labs can be licensed there and people can be trained to develop all the reagents and all the technology can be transferred, if this is possible.
On my side of the project, I think this is possible, yet a bit challenging, because the technical competence may not be present in all countries. Our countries might have it. It's a bit difficult to say that. But what we know for sure is that given a bit of a technical input, given the protocols, and given some trainings, which in these days have to be online because there's no travel possible in these times, it should be realistic to set up something within the given time. And I mean, as I said, that I've been approached by private investors from India, which is the country where I come from, and also from other healthcare companies who are trying to market this product or want to market this product in India and Asia, for example. So there is a lot of interest, but as I said, that we are still trying to figure out what is the best strategy to reach out to as many as we can. We have also discussed in depth of opening up a production facility that can handle all the, at this point, we are enough to handle the whole of Norway, that's for sure. And then we are talking about neighboring countries where you are currently there in Vakas, for example, Sweden, Denmark, etc., where they need. And that can only happen once this approval from the health agency comes in, because then we have a good background, let's say, to approach and to offer help to people or countries who would need them. Coming back to Pakistan, India, Bangladesh, and the region there, I think it's very important to understand that these kind of equipments would be available in several of the biotechnology or biolabs and several other institutes within these countries. So it is only to train these people to do a specific work. And as you know, I mean, the number of people in India or Pakistan is not a problem. I mean, they can work if needed for hours, but it's only the what we need to solve at this point is how we do this transfer, what is best, and if there are interested partners who would take it forward without looking more from a commercial perspective, but for serving society in the situation of crisis. That's what mine and Magner's initial aim was, to be a part of this project to help people fight this coronavirus.
That is very encouraging to hear. So I hope that somebody is listening, and they can approach you guys and actually get training for the people and set up this kind of lab to get this type of testing done. Because otherwise, we know that as we keep reading in the newspapers that the tests are in very short supply in Pakistan, as we know, and of course it should be in short supply in India as well, and the neighboring countries. So I hope something in this regard happens and things get better. If you guys have any other questions, Asif or Atif?
Yeah,
Shulalit,
Shulalit, as you have mentioned that health agency approval is required first to move it to other countries. So how much time you are expecting that it will take for the approval from health agency from now?
Yeah, I mean, I do not know the exact time, to be honest, but I know that before Easter, the documents have already been forwarded to the health agency, and that they are working on it. And I think once after Easter, we should be able to get some indication of how long it would take. But I'm not guessing in months, but it would be in a week or so, because Norway, first and foremost, needs testing at this point. So they are very aware of this method being developed and being tested already and validated against other existing protocols. So I guess it would be somewhere towards the beginning of next week. But this is my guess. I don't know how the authorities are doing. It could be even earlier, given the situation.
Yeah, thank you very much, Lalit, for the insight.
Thank you.
Atif, you have any question?
No, I just want to say a big thank you to Mr. Banerjee for sharing all the details about his new way of testing the patients and everything that involves it. So it was really nice to hear that, and hopefully things would actually get better and people would benefit from his technique of testing as well, inshallah.
Yes.
Especially the people of Pakistan and India, the
Yes, I
area
hope
that he
so.
comes from.
Thank you.
That would certainly be great. Hope all of the humanity benefits from this. Anyway, Sulalit, we won't be holding you for long because, as we know, these days your time is much more important than ours. So tell us about production. Is it going at full pace now, right now?
Yeah, we haven't. So the last week we have been mostly setting up facilities and training operators. So now it's a total team of 7 people on our side, on the production side of magnetic beads, who are all trained and who are doing shifts, and we are working 7 days a week running these facilities. And from this week, we have started to produce at full capacity. So, as I mentioned at the start, we are now able to produce 60,000 tests with per day, which we think is quite huge from what we were producing initially 2 weeks back, which was 1,000 tests a day. So we have gone, you can do the math, 60 times more or scale up, and that has been challenging, because as chemical engineers all discussing now, you would know that scale up is an issue in anything you do. So we are very happy that it kind of worked out in this short time, and with the efforts put forward by the whole team and a lot of support stuff. I must acknowledge the support that we have got from the department and the faculty by providing facilities, speeding up processes which would otherwise in normal circumstances take quite a lot of time, giving overnight access, discussing and having meetings late in the evening. So these have been very, very helpful. So everybody has come together to kind of support and promote the activity that we are doing.
That's great to hear. And I wish you good luck in the scaling up process.
Thank you.
And may God give you the strength and stamina to sustain this kind of production. So here I will end this podcast. Thank you very much, everyone, and thank you especially, Sulalit, for taking time out to tell us about this kind of testing, and hopefully it serves the humanity, as you have mentioned on various occasions already. So that was all from us. Thank you very much. Bye bye. Take care.
Thank you.